This is restriction in the nuclease BAM-H1. It binds DNA at specific sites and breaks it into two parts with sticky ends. We can use this to create multiple of these fragments, to break them up and piece them together as we please. But how do we piece them together? Well, we can use some sort of tape, but that won't work as we want the DNA to be exactly as before. What if we could bring them closer and rebuild the bonds between the DNA strands, in effect making them one? DNA ligases are the cellular machinery that stick together two DNA strands that are otherwise disjoint from each other. They are crucial for DNA replication and repair, as you would see later in the video. There are broadly two types of DNA ligases, one which use ATP as an activating agent and are primarily found across eukaryotes like us humans, and the other which uses NAD+, which is seen in prokaryotes like E. coli. Our main focus of this video would be the ATP-dependent DNA ligases, and we'll be studying the mechanism and function by having a closer look at human DNA ligase 1. Let's first take a look at how cells' repair machinery works. One ATP molecule binds to the protein, causing a conformational change, which allows the protein to bind to the DNA. Through a series of bond breaking and making as shown here, the adenosine monophosphate moiety ends up bound to the 5' end of the broken DNA. This destabilizes the molecule, favoring an attack from the 3' hydroxyl group, which then reforms the phosphodiester bond of the DNA, releasing AMP and the enzyme back to the open to act somewhere else. Ligases have proved themselves useful in many places both inside and out of the cell. Let's look at the few examples of the same. During DNA replication, DNA polymerase, the enzyme responsible for assembling the complementary DNA strand, works in a unidirectional manner, always reading the DNA from the 3' to the 5' end. Due to this, one of the strands is made at a stretch while the other has to be made in small segments as shown here. These segments are called the Okazaki fragments, which are then joined together by DNA ligase 1. DNA ligase also helps in repairing DNA damage. Most of the damage done to the DNA is in the form of a mutation or a bond breakage. The former is taken care of by multiple proteins along with ligases, while the latter is fixed by ligases alone. We can see that the job of making the phosphodiester bond is an irreplaceable one. During experiments, we more often than not want to add a gene of interest inside a plasmid so that the bacteria can produce it. This is done by cutting both the plasmid and the gene with the same restriction in the nucleus so as to produce the same sticky ends, and then using DNA ligases compatible with prokaryotes to join them both together.